The next speaker is Dr. Rui Xu from Beijing University of Posts and Telecommunications, and he will speak us on the topic of functional fingerprinting for the developing brain using deep metric learning and ABCD study. Let's welcome.
Hello everyone, my name is Rui Xu. I'm from Beijing University of Post and Telecommunications. The topic of my talk is functional fingerprinting for the developing brain using deep metric learning. I confirm that I have no disclosures. There are significant individual differences in the brain functional networks. As you can see in the picture, the individual differences in the adults are substantial and are even greater in children. The questions people are always curious about is how to characterize the unique developmental course of brain functions. Brain functional fingerprinting is an important way to study individual differences. It captures unique connective patterns for each individual. Studies have shown that brain fingerprints are distinct and reliable for individual identification in adults. However, characterizing children's fingerprinting remains challenging, but they are changing fast. Considering fingerprinting technology, traditional methods are poorly based on the brain connectome, while deep learning offers a new way by removing shared features and highlighting individual patterns. However, in previous methods, brain dynamics are largely ignored and understandability remains limited. So in this study, we aim to develop a brain fingerprinting framework based on brain dynamics and explore the unique brain functional patterns during the development. We included 1,325 children from the ABCD study. This data have at least one resting state fMRI session across 3 time points over 4 years. Each session contains 1 or 2 runs. All data were pre-processed by Kato. Finally, we got a time series 219 brain regions with a length of 375. We developed the magic boat model for brain fingerprinting. The model takes 3 metrics of time series as input. 2 data metrics are from 2 runs of a same individual. The third one is from a different individual. The model employs a transformer block to encode a fMRI time series into brain fingerprints by capturing both local and global temporal patterns. And we adopt a metric learning approach here and use the triplet loss function. This ensures that brain fingerprints from the same individual are close together, and those from different individuals are far apart. After training the model, we can use our model for individual identification. For this, we need to first build a database of fingerprint from all subjects in one run. Then in the second step, we can compare the fingerprint of a target individual to the database. We only need to look for the individual with the smallest distance. Here we show the main results of individual identification. As you can see in the picture, we got a high accuracy of around 97% for identifications within a session. We also got a high accuracy of 92% for data spanning 2 years and 87% for data spanning 4 years. These results are better than the functional connector approach. We also interpret our approach by examining which brain regions contribute most to the brain fingerprints. Notably, bilateral middle temporal regions played an important role in individual different identifications. We further used the neural maps and found that those important regions are mainly involved in higher-order connector networks, especially the default mode network. Notably, the patterns of regional contributions resembled to the inter-subject variability map and the SA axis. Once more, we examined the relationship between the brain fingerprints and genetics. We found that genetically related individuals, for example, twins and siblings, showed more similar brain fingerprints compared to genetically unrelated individuals. We also used the whole brain gene expression data from the OHBM to study which genes showed similar patterns with the regional contributions to fingerprints. We identified a couple of genes and showed these genes to be associated brain development during late childhood. We developed a normal deep learning framework, metric bot, to study individual differences. The framework identifies unique brain functional fingerprints in the developing children. The most distinctive brain fingerprints are driven by higher order association regions, especially the default mode networks, and are influenced by genetics factors. Finally, I would like to thank my supervisor, Professor Yongbin Wei, and all other co-authors of my work. And you can also find me at my poster. My poster number is 1014. Also, you are welcome to other posters from our lab. Thank you very much for your attention.
Thank you, Rui, for the very nice talk. There are a couple of questions on the app, so I just pick maybe 2 of them. The first one is, since your study is done in the ABCD population, so how does this model perform on the adult population? Have you tried that? did you try your model on the adult data sets?
We included 1,325 children from the ABCD studies.
So you haven't tried that on the adults, right? Like adult population. It's okay, we can move to the next one. So what is the difference between brain fingerprinting and normative modeling?
The functional fingerprinting is the unique functional patterns of FMR time series.
Right, so in my understanding, like the fingerprinting, the network you present here is one way of capturing the features of brain development. So how does it compare to the normative modeling approach, like a more conventional approach?
We developed a metric bot model to encode time series into fingerprints.
All right, then how does individual signature change with brain development in your data set? Like the key features captured in your network, how does it change with development?
This model takes three time series as its inputs.
Okay, since the time is up, so let's thank the speaker once again.